All right, we're here to talk about DNA being known as the actual thing that is creating or passing the genetics on from organism to organism. So let's get through some of this stuff. In the late 1800s, early 1900s, scientists were really trying to figure out, is DNA actually the stuff that's causing heredity, causing the passing on of information from parents to offspring? So in order to lend support to these ideas, scientists had to come up with some circumstantial evidence, and they came up with some experimental evidence. So let's look at some of these things and try to figure out how we know DNA is actually the genetic material. Well, first thing that we need to look at is what conditions need to be met for us to say, yes, we're sure it is. That's what we're looking at. Well, the first thing we need to do is figure out if DNA actually is in the nucleus, and it actually is what makes up the chromosome. So we're going to see experiments that show that. Does DNA double? Is there more in a diploid cell? And is that actually the stuff that's providing the information to cause a change in an organism? So these are the 4 things we're going to kind of check off. Let's go through some of these experiments and evidence that came about with scientists. The first thing is Friedrich Miescher. He was looking at bandages of wounded soldiers, and as he was looking at it, he noticed some fibrous stuff, and he analyzed the fibrous stuff to try to figure out what elements were found in there. Well, he found a few specific elements that, when we talk about DNA, they're really kind of important. CHO, we've seen those elements before. Those are in carbohydrates, and if you remember the base structure of a nucleotide, which is an individual monomer of the strand of DNA, one of the components of a nucleotide is some kind of sugar. So CHO would absolutely fit that bill. Another thing we find in a nucleotide is a nitrogenous base. Well, there's some nitrogen that he found inside this fibrous stuff that he kept looking at. And the last thing is a phosphate ion, and look at that. It's carbohydrate, carbon, hydrogen, oxygen, nitrogen, phosphorus, all found in this fibrous stuff that he called nuclein, because it all came out of the nuclei cells that were found in these soldiers' wounds. So now we're starting to get the idea that DNA is present. It's present in the nucleus, and it's present in the chromosomes. Now, around the turn of the century, we had the Industrial Revolution going, and dyes were becoming more and more popular for scientists, for the general public as a whole, but for scientists also. They started taking these dyes, and they're like, what can we do with this? Well, they started to put it onto cells that they're looking at in the microscopes. Microscope technology is getting a little better, and these dyes are becoming more affordable. And they found out that there's certain parts of the cells that stained really, really well. They called these things the chromosomes for colored bodies. That's what chromo and some means in the Greek and Latin roots. So as we can look at these chromosomes, now it's a lot easier to count the chromosomes. So now we can figure out, does the amount of chromosomes actually change throughout the course of the cell cycle, and is there more DNA or chromosomes in one cell rather than another? I'm checking somatic cells compared to gametes. And now that the chromosomes are colored, they can figure that out pretty easily. The next thing happened when scientists started playing with viruses. And if you look over here on the right, that little thing that kind of looks like a space monster is what we call a bacteriophage. Bacteriophages are viruses that attack bacteria. And viruses are kind of weird little organisms, because what they do is they just are made of protein and DNA, basically. And they are not really alive unless they attach to a host. And when they attach to a host, they insert their DNA into that host, and basically that DNA gets incorporated into the host DNA, and that cell that the virus is attached to will start making little virus parts, and then once it makes enough virus parts, the cell dies and viruses escape, and now you got more little baby viruses, even though they're not really alive because they don't do any life processes. They just are sort of carrier things. Well, we found out by tagging the protein and tagging the DNA that only the DNA was going into the cells. And if that's the case, that means it's only the DNA that's causing the change in these bacteria cells to make the virus parts. It had nothing to do with the protein. It was simply the DNA stuff inside the virus. Well, now we're coming up with some pretty good experimental evidence in addition to all the circumstantial stuff. Those experiments where you change the genome of an organism by inserting DNA from something else into it are called transformation experiments. And anytime an organism takes in the DNA and is transformed, we call that organism transgenic. So let's look at our 4 conditions. Did we find that DNA was present in the cell nucleus and chromosomes? Check. That was Friedrich Miescher that found that in the wounds of the soldiers. Were we able to count the DNA during the S phase and compare diploid cells to haploid cells? Check. We did that when we were able to dye things. Did we see some transmission of genetic information? Yeah, absolutely. We checked that when the bacteriophage was killing the bacterium. So now we've kind of checked all our boxes. So now scientists are saying, yeah, DNA is the genetic material. So what does DNA look like? Well, now we know it's a genetic material, we need to figure out a couple other things. How does it replicate, and how do proteins come from DNA? How does this whole process actually occur? Well, first we need to figure out what's going on and what it looks like, because if we know what it looks like, that kind of unlocks, once we know what it looks like, I should say, that kind of unlocked all of this information that tells us how DNA is replicated and how protein synthesis takes place. Rosalind Franklin was a scientist out in England. This is right after World War II, and she was working with Morris Wilkins. Being a female, she had to sort of do her thing on the slide, had to work under somebody, but she was really good at using x-rays to put them through molecules, and as the x-rays pass through molecules, they diffract off in different directions. Well, depending on how they diffract off and move and get deflected, they create an image, and she put x-rays, put it on a photo plate, and she was able to analyze these photo plates with all these little black dots that you see over here on the left, and by sort of doing some math calculations and going backwards, she could figure out where all these atoms sort of where they are because of the way that the DNA is turned up, the way that the x-rays, are deflected out onto the image. Well, that was kind of a big deal. What she didn't know is that Morris Wilkins sent her information over to some other scientists, and we'll get to that in a second. But when we look at the nucleotides and the DNA itself that the x-rays are going through, hopefully we remember that nucleotides have that phosphate, the sugar, and the base. When we look at our nucleotides, the bases, there's 4 different bases in DNA. We have adenine, thymine, guanine, and cytosine. They follow Chargaff's rule, and this is one of the things that Franklin sort of found out by passing these x-rays through all the strands of DNA. She always saw the same pattern, and this idea that A always pairs with T, C always pairs with G, is what we call Chargaff's rule, and you should know which ones are pyrimidines, which ones are purines. If you use the acronym PUGA2, the purines, PU, are guanine and adenine, G and A, and then the 2 refers to the number of rings in the adenine and guanine molecules. So if you can remember PUGA2, you can remember which bases have 2 rings and which ones are purines. Well, Watson and Crick got a hold of Franklin's x-rays. Morris Wilkins actually gave them the x-rays, and when they got a hold of them, they were like, wow, this is some really big stuff right here. This is huge stuff. And what they did, they took one piece of paper, wrote down their findings that they got from Franklin's x-rays, submitted them, they got published, and they're credited with discovering the structure of the DNA molecule, the double helix structure. And basically what they said was that the bases are always on the inside, and the backbone is created by the sugar and the phosphate. So those nitrogenous bases point to each other, and they match up and are held together with hydrogen bonds following Chargaff's rule. Huge stuff right there, because once we figured out the structure of DNA, and there's some really big stuff about the structure that we'll talk about in a second, but that structure and understanding how DNA is oriented sort of unlocked a whole field of genetics for scientists. So let's look at this structure. DNA is what we call a double helix because it kind of looks like a ladder that's twisted. When you look at one strand of DNA, it's actually 2 strands. When you look at this one double helix, it's actually 2 strands of DNA. Now you'll see some numbers up at the top, and at the top of this one, you see a 5 with an apostrophe and a 3 with an apostrophe, and we'll talk about that 5' and 3'. When we look at DNA, we say that it's a right-handed double helix, which if you follow the backbone from the 5' end down or from the 3' end down, and you pretend you were walking downstairs, it's almost like you're constantly turning right, and that's sort of what this right-handed double helix means. The only sections of DNA that are left-handed are something called zDNA, which we don't really go into in this class, so I'm not really going to worry too much about that stuff. Some big stuff about DNA, and this is kind of the most important thing, DNA is anti-parallel, which means DNA runs in the 5' to 3' direction. That sort of start is 5', the 3' is the end. And when we look at a picture of a nucleotide in a second, you'll see what I mean by the 5' and the 3'. But If I have an A on one side, I'm always matched with a T. If I separated those two strands of DNA and I have A on one strand and T on another strand, well, when I match those up to make a double helix, they're going to make exact copies of each other because they follow Chargaff's rule. And as they do that, they create these things called major and minor grooves. And that has to do with spacing between the backbone on either one side or the other. The major and the minor grooves add kind of checkpoints along the double helix of DNA. I always compare them to mile markers on a freeway, where if you know that you're at mile marker 32 and you get in trouble, you call a police officer and say, hey, I'm stuck at mile marker 32, and they know right where to send stuff to you. On these major and minor grooves, depending on how they're arranged throughout the strand, that's going to add locations for things to grab onto the DNA. Maybe a protein, maybe an enzyme, maybe some kind of methyl group, something like that can grab onto it by finding the right major and minor grooves. Now, when we look at the nucleotide, we see the phosphate on the left, the sugar right in the middle, and the nitrogenous base on the right. You'll see the numbers next to all the carbon atoms, and hopefully you remember the corners on that molecule are all represent carbon atoms. Well, you see 1, 2, 3, 4, 5. The 5' carbon is always the one that's right next to the phosphate group. The 3' carbon is always the one that's opposite the oxygen. When we say DNA runs from the 5' to 3' direction, that has to do with the orientation of the sugar in the strand. Well, if 5' is on one side, on the opposite strand that's matched up, let's say you had an A in this nitrogenous base, it would be matched up with the T, that would be the 3' end over there. And we got to look at this in class, and you can see it on this picture. You can sort of see. You have the 5' end on the top right, you have the 3' end on the top left. And look at the sugar groups. They're upside down. That's what we're talking about with anti-parallel. So the A's are always with the T's, the G's are always with the C's. The A and the T make two hydrogen bonds between them to hold them together. The G's and the C's make 3 hydrogen bonds to hold them together. That's stuff that we should know. And we could see that the sugar and the phosphates just sort of orient themselves the right way. So because of this double helix, there's 4 big things that the double helix allows. First off, it's a storage of genetic material in that sequence of bases. Second, by splitting those and following Chargaff's rule, replication becomes very, very precise. Third big thing with the double helix is that it is susceptible to mutations, which don't assume mutations are bad. That can be very good because it can lead to genetic variation, which in this class, genetic variation is always a good thing. The last thing is that it allows information to be expressed as phenotype through the synthesis of proteins. That's all we got for DNA structure and sort of the background of DNA. We will get into the replication process next. Have a good afternoon.